There are an amazing number of different causes for chronic wounds, including diabetes, venous insufficiency, pressure sores, surgical site infections, and many more. Yet none of these causes are sufficient to explain the biochemistry and cellularity found in all chronic wounds. Chronic wounds are stuck in a persistent inflammatory state. Biofilm, a surface-associated society of bacteria, is the best explanation for this inflammation and is now recognized as an important barrier to wound healing. Biofilm gets its start when bacteria gains access to internal host tissues through a breach in the skin. Usually single-cell bacteria, planktonic bacteria, are seeded onto the host tissues. These planktonic bacteria have multiple symptoms to identify an internal host environment, such as heme sensing systems or receptors to specific epitopes of the extracellular matrix. These systems are yet unidentified mechanisms that cause the bacteria to twitch until they irreversibly attach, which is the first committed step to biofilm formation. Attachment of each bacterium stimulates receptors, which progresses through signaling pathways, which produces the radical metamorphosis of an individual bacteria into a biofilm society. Quorum-sensing molecules, receptor signals, even host molecules work through secondary signaling molecules, mainly a two-component system, to exert epigenetic control over the DNA of the cell. This activates operons, or suites of genes, to be expressed. In fact, up to 800 new genes are expressed in the metamorphosis of a planktonic bacterium into a biofilm phenotype. These newly expressed genes result in over 50% of the outer membrane proteins of the bacterium changing. Also, extracellular polymeric substances, such as proteins, DNA, as well as the polymeric sugars, are secreted. But more importantly, the effector molecules that are produced by this gene expression are released through T3 secretion systems as well as T4 secretion systems. These effector molecules are utilized by the biofilm community to usurp many host molecular pathways that are commandeered by these effector molecules to produce a hyperinflammatory state, which will nourish the biofilm and prevent host healing. For example, these effector molecules can block apoptosis, thus immortalizing the cells and tissue that the biofilm is attached to in the host environment. This simple strategy prevents the host from lysing the surface and sloughing the biofilm. The early biofilm, a microcolony, is now fervently attached to the host surface, and each member is secure within the self-secreted matrix. The bacterium of the biofilm society are now resistant to host immunity, such as white blood cells, antibodies, and complement. Also, they are resistant to antibiotics, biocides, and many other agents. As the microcolony continues to mature into a formal biofilm, quorum-sensing molecules direct the protein expression within each bacterium, causing it to differentiate into functions for the needs of the community. Phenotypic diversity, along with genetic diversity, that is, multiple different species of bacteria within the biofilm, allows the biofilm to reconstitute itself after a major attack. This is an important survival mechanism for biofilm. The mature biofilm's other defense mechanisms include slow penetration of toxic substances such as antibiotics and biocides, accelerated horizontal gene transfer in the mid-portion of the biofilm, and then in the deeper regions, very slow-growing or metabolically inactive bacterium due to toxic waste products and low nutrients such as oxygen. These colony defenses, along with colony virulence factors, allow biofilm to cause human infection quite different than anything we have ever known.